Hello everyone, and welcome to the first live session inaugurating the e-conference entitled Cell-to-Cell Metabolic Crosstalk in Physiology and Pathology. This conference has been organised by MDPI and the journal Cell. I would like to thank all the speakers and all the participants for joining us today. The conference dates are actually from the 17th of December to the 17th of January 2021. Over that period, you will be able to read and comment all the authors' submissions presented for the conference. You will also be able to enjoy some live sessions starting from today and up until the 18th of December. Please look at our webpage for more information concerning the abstract submissions and the live sessions. I would also like to remind everyone that this webinar is being recorded, as well as being live streamed on YouTube. The recording will be posted as soon as possible on the webinar's webpage for everyone to watch. Today, there will be two presentations that are both part of session B, Exosomes and Extracellular Vesicles in Health and Disease. The first speaker is Professor Dr. Jan Lötvall, who will give a talk on extracellular vesicles as communicators between cells, importance in health and disease. The second talk is given by Dr. Dhanu Gupta, who is standing in for Dr. Samir El-Andalusi, who unfortunately had prior engagements and could not be present at the conference. The talk will cover the topic of the engineering of extracellular vesicles. Each speaker has 30 minutes and there will be, or maybe not, a 10-minute break between both talks. And now I hand over the microphone to Professor Lötvall. Thank you and enjoy.
Thank you very much. It's a great pleasure to join this session. I do apologize for the delay. Is the microphone okay? Does it sound good? Thumbs up?
Perfect. We can hear you.
Thank you very much. So my name is Jan Lötvall. I'm a professor at University of Gothenburg in Sweden. I actually have a background in allergy and asthma research, but around 15 years ago, we started working on something that was very new at the time called exosomes, and we have not stopped since. So when you look into human tissues, and this is a human bronchial biopsy tissue, you can see a cell here. I'll get the pointer active. You can see a cell here with a nucleus, and you can see collagen fibers, but you can also see a lot of circular structures around the cell in the interstitial tissue. In this case, the cell also seems to be vesiculating and discharging a number of vesicular structures on the side. So if you look with a little bit of higher magnification away from the cell, you can continue to see some of the collagen here and here, but you also see these circular structures throughout the tissue. Many of these are quite big. These are multiple hundreds of nanometers, maybe a micrometer in diameter. But if you look with even higher magnification, you can see vesicles or round structures that are down to 50 nanometers, which are what is considered to be a size, a diameter of an exosome. So as I mentioned, my name is Jan Lötvall. I'm professor of allergy. I have, this is my declaration of interest, with multiple patents of using exosomes as therapeutics and diagnostics. I have worked with a company called Kodiak Biosciences, which is a biotech company pursuing exosomes as therapeutics. I've started another company called Exocure Biosciences. I'm currently editor-in-chief for Journal of Extracellular Vesicles, and I'm the past president of the International Society for Extracellular Vesicles. So just want to acknowledge the amazing team that I have around me. A number of people that have been very important in the work that I'm presenting today is Cecilia Lesser, who's been in the lab since the very early days of our exosome research. He's done no other type of research really. Kyung-Soo Park, who was trained by Yong-Sung Go in South Korea. Yong-Sung Go, we started collaborating in 2012, and that was very, very helpful. And also Rosella Crescitelli and Alexander Svietkovich have been very crucial in this work, as well as Suchul Jung, also from South Korea. So when you look at cells, they have the ability of releasing very many different types of extracellular vesicles. And those vesicles can mediate surface signaling. They can be taken up by a recipient cell and they can deliver, for example, proteins, but they can also deliver RNA. And this is where our lab came in early, showing that exosomes can shuttle RNA from one cell to another. So, but when you look very carefully into the vesicular secretomes, the vesicles that are released by different cells, there are these vesicles that are released from the surface of the cell that are commonly called microvesicles. And those that are made inside of the cell in multivesicular endosomes that are called exosomes. But if you start looking carefully into these populations that are isolated with slightly different methods, using ultracentrifugation and other methods, you can see that there is a diversity of these. They're not all the same. They are slightly different, and there seems to be subgroups. That's true for the larger vesicles that you collect at 10,000 G centrifugation, but it's also true for the exosome preparations that you isolate at 100,000 G preparation. So it seems that these vesicles have the ability of conveying multiple types of biological functions between cells, including surface signaling. They can deliver different molecules, and there are many different types of vesicles seemingly released by a single cell type, and they may have different functions, and the relative concentration of those vesicles may vary under different physiological circumstances. So, so the question is, are there then fundamental different subgroups in vesicles released by different cells? And that's something we've been pursuing over several years. So this is a figure that is coming from a review article we published 2 years ago, where we look at the evidence that argues that there are subpopulations of extracellular vesicles. I mentioned in the previous slide that you can isolate vesicles at 10,000 G, and you can isolate vesicles at 100,000 G, and these are commonly called microvesicles and exosomes. And when you look carefully at the difference between these, you can see different types of cargoes. There are also differences in the biogenesis of vesicles, and I mentioned the exosomes are produced in the multivesicular endosomes, whereas microvesicles are produced by budding from the surface of the cells. Now you can also look at morphology, and I'll show some cryo-electron microscopy that describes multiple types of morphologies in extracellular vesicles. And of course, size, and there are different ways of isolating vesicles with different sizes, including size exclusion chromatography, but also this sedimentation method with 100,000 G that isolates smaller vesicles, and the 10,000 G that isolates bigger vesicles. So these are connected. But you can also start looking into the subpopulation of vesicles, looking at their protein or molecular cargo. So seemingly different types of vesicle may have different surface molecule, and that may be very fundamental and important component of determining their functionality and their phenotype. And also we can find both DNA and RNA cargoes in the extracellular vesicles, and that may also describe or identify different types of vesicles, different subpopulation of vesicles coming from a single cell type. So let's start with the morphology. So here, this is a cryo-electron microscopy number of cryo-electron microscopy pictures from vesicles released from a single cell type. But this is produced by Johanna Hög, who's an excellent electron microscopist. And you can see here you have multiple types of different vesicles. If you look at these three figures, these three photographs up here, you can see the round structure here, and what you can actually visualize with this technology is the membrane. So you don't need any stain when you use cryo-electron microscopy, but you can visualize the lipid bilayer. And most of the vesicles, when you look into the cryo-electron microscopy, this is basically taking the supernatant, concentrating the vesicles and freezing them, and then immediately putting them in the electron microscope. You can see these single vesicles, they dominate. They're the most common vesicles that you find in this situation. Now, you can also find double vesicles that have two membranes. So here's one outer one and the inner one, and there seems to be two vesicles, one vesicle inside of another vesicle. So these are also quite prominent, but they're also vesicles with multiple small vesicles inside, and all of these have slightly different morphology, phenotypes. Also, you can find small double vesicles, you can find oval vesicles, you can find small tubules that are quite short. You can find pleomorphic vesicles that we can't describe other than that they're looking quite different. And then we have some long tubules. And sometimes you have these really long vesicles that seemingly have actin filaments inside of them. And we have some data to show that these can actually have some ability to move in vitro. So here is, let's see if we can get this up and running. Not sure that's going to work. Of course not. But these vesicles can actually extrude their different membrane components and seem to have actin filaments inside that are responsible to this movement of the extracellular vesicles. These vesicles are usually larger than what we normally talk about, which are the exosomes or the smaller vesicles. But these can be found in all types of vesicle isolates. So if you look at if you try to characterize the different vesicles, you can find these 6, 11, 16 different types of vesicles that you can find in the secretome from any type of cell. And this is perhaps one way of characterizing subpopulations purely by morphology. So what you also can do is to then look at the size or the density sedimentation. I mentioned these are isolated with 100,000 G and they're smaller, and these are larger and isolated with 10,000 G. And that was the most common way of isolating vesicles if you look back 10 years and beyond that period. And what is important here is that these vesicles actually seem to have different nucleotide cargos associated with them. So this is a paper that we published in 2007, which showed that exosomes carry a lot of different RNAs, both mRNA and microRNA. And that RNA in the exosomes is fundamentally different from that in the cell. And here we can see that the exosomes have much smaller RNAs compared to the cell, and they do not have the ribosomal RNA. Furthermore, the exosomal RNA does not correlate with the cellular RNA, so there seems to be fundamentally different. There is some sort of loading mechanism of the vesicles of the different RNA cargo components. Now, if you look at the 10,000 G palette, and this is from my colleague Yong Song-go in South Korea, published only a few years later, they showed a very close correlation between the vesicular RNA and the cellular RNA regarding mRNA here. So they see a correlation, we did not see a correlation. And the big difference here is that they are working with microvesicles and we are working with exosomes. So exosomes, there's no correlation. Microvesicles, there is. And also in the microvesicles, you see these ribosomal RNA peaks that you do not see in the vesicles, in the exosome vesicles. So clearly there are different RNA cargos in vesicles coming from different or being produced by different ways, or isolated by different ways. So we further pursued some RNA deep sequencing to distinguish differences between subpopulation of vesicles, and this is work by Tara Lunavat in my lab. He's now in Norway, but he was very diligent and isolated exosomes, microvesicles, or apoptotic bodies, which in some cell cultures can be a significant portion of the vesicular secretome. So those vesicles are produced during programmed cell death. And then he compared with the cells. And when you look at the heat map here, clearly the exosomes are very different from the microvesicles, but also the microvesicles are different from the apoptotic bodies, and the cells have their own RNA-seq profile. And when you look at the principal component analysis, you can see that the apoptotic bodies, the microvesicles, the exosomes, and the cell actually distinguish from each other. So all of those components actually have different RNA cargo inside of them. So we've also been pursuing quite extensively and more recently the characterization of the surface proteome of extracellular vesicles in an attempt to trying to identify subpopulation of vesicles using that principle. So if you think of vesicles, they do have this cell membrane, but of course they do have different membrane proteins, cargos on their surface. But if you look at the crude isolate of extracellular vesicles, you can find thousands of proteins, and around maybe 700 of those proteins are actually membrane proteins, but also many, many different RNA species. So we're trying to understand whether we can capture or identify vesicles that have distinct surface molecules. So when we did some membrane proteomics, and this is work pursued by Suchul Jung when he was in the lab, he's currently at Kodiak Biosciences in the USA. You can see here that that you find a lot of in the bioinformatics, you can identify a number of vesicles that are, or this is percentage. A large percentage are plasma membrane proteins coming from the surface of the cell, and that makes sense. But you can actually find 27% of them have endoplasmatic reticulum membrane proteins, and 26% of them have mitochondrial membrane proteins. And this is not actually any mitochondrial membrane proteins. It's specifically and almost exclusively mitochondrial inner membrane proteins. So it's not the outer membrane proteins that you actually see. So what we did was to identify a unique mitochondrial inner membrane protein that seemed to be prominent in these vesicles, and we developed a bead with an antibody binding that MTCO2 mitochondrial inner membrane protein, and did proteomics on those vesicles. And when you enrich those, you can dig much deeper into the proteome, and you get a number of unique proteins. So that's clearly arguing that there is a subpopulation of vesicles that carry the MTCO2 molecule. What we also can see is that we have some sort of mitochondrial enzymatic activity in this situation. So this is the relative ATP synthase activity in just general exosomes or the MTCO2 enriched extracellular vesicles. So it's been known for probably more than 40 years that there is ATP synthase activity in extracellular vesicles, especially in what is called prostasomes that are produced in the prostate. But here we can show that these mitochondrial inner membrane vesicles are enriched in these in this enzymatic activity. So we've also developed different analytical tools to try to quantify these different subpopulations. And for one instance, we did a direct ELISA where we look at for specific single proteins by binding the vesicles to the surface. And when we do that, we can identify a number of different surface molecule on the different vesicles from different sources. And here I shift to a slightly more clinical approach. So here we have actually vesicles that are from melanoma patients. So these are from metastatic melanoma. And what we do is we isolate the vesicles from the tissues in that case. And here we have a melanoma cell line. Here we have a mast cell line, an inflammatory cell line, and here we have mesenchymal stem cell vesicles. So we put these different vesicles on the plate, and then we look for these different surface molecules. And when we look for this molecule, which is a mitochondrial inner membrane protein, COX-6C, we can see that it is present in the vesicles from the tumor tissues, but not necessarily from the cell cultures. SLC25A22, the same thing. MTCO2 was interesting because we could measure that also in the cell lines and in the inflammatory cell lines, but not in the MSC. And I can also say that we cannot find these vesicles in, for example, in HEC293 supernatants. We looked at a plasma membrane and the plasma reticulum and of different types, and we could confirm that we could find those vesicles as well in the tumor tissues, but less so comparatively in the different cell lines. To make a long story short, we ended up with developing an ELISA where we looked for vesicles that carry both the COX-6C and the MTCO2 surface molecule. And then we developed a sandwich ELISA for these molecules. So this is the MTCO2 antibody which we plated to the plastic. Then we put the EVs in, and then we put a secondary antibody to, for example, the mitochondrial protein, the plasma membrane, or the endoplasmatic reticulum. And this is a little bit more complicated data, but what you can see here, if you look at for the MTCO2 and COX-6C double positive vesicles, those were significantly increased in the tumor tissue vesicles, but they were not measurable in the mast cell line or the mesenchymal stem cell supernatant. So if you combined MTCO2 with some other marker, for example, CD9, we could not see differences between the different types of sources of the vesicles. And here you can actually measure the CD9 and MTCO2 vesicles in the mitochondrial membrane, in the mast cell line that supernated. And MTCO2 and plasma membrane, very difficult to measure. And mitochondrial MTCO2 and the plasmatic reticulum, we could not identify. So we thought this was an interesting combination to look further with. So here we extended the work and we looked at 5 different melanoma metastases where we again took the tissue and isolated the extracellular vesicles directly from the tissue. And here you can see that they were measurable, clearly measurable in all of these tissues. You can measure them to some degree in the mast cell line, very little so in the MSC supernatant, and they were not present at all in the vesicles that are produced by HEC293 cells. So they don't have this subpopulation of extracellular vesicles, seemingly. So what we started to do is to think whether or consider whether this could be useful for diagnostic purposes. So specifically, we choose to use the MTCO2 and the COX-6C combination rather than the MTCO2 and CD9, because this seemed to be more prominent in the tumor vesicles. And when we actually look in the plasma And this is looking at unpurified plasma, just plasma, and we put the plasma onto this assay, and we put in this primary antibody and the secondary antibody. We can measure these vesicles in healthy blood. So all of us, all of you listening here, have these vesicles circulating. But what was interesting here is that we could find an increased concentration of these empty CO2 coccyx-C positive vesicles in the circulation of those with metastatic melanoma. And we're not saying that this is a diagnostic tool, but perhaps it can be a candidate for diagnosis or understanding what the tumor burden may be in these instances. So I think this is describing one of the first descriptions of a subpopulation of extracellular vesicles potentially being useful as a diagnostic tool. But of course, there are many other opportunities of looking for diagnostics in extracellular vesicles, and you could look at cargo proteins, you could look at different RNAs. And what's very popular now is to look at mutations in the RNA. So there are companies out there looking for that specifically, and also in extracellular vesicle associated DNA, looking for oncogene mutations, for example, in that component. So in summary, all cells produce different types of extracellular vesicles. Some of those maybe are not important for physiology, and some seem to be very important for cell-to-cell communication. We don't understand the relative distribution of these subpopulations being released from different subtypes of cell, but clearly that's a very important research to be pursued. And we can here see that the subpopulation of mitochondrial inner membrane protein, not outer membrane, inner membrane protein, is described in this instance. Maybe it can be diagnostic, but there are many other possible subpopulations that may be discovered in the future. Thank you very much for your attention, and apologize for the delay in the start.
Thank you very much, Jan. Very interesting. I don't know if there are any questions. I can wait a few seconds to see if people have questions for you. And if not, we can pass on to our next speaker. Well, they seem rather quiet. So all I can say is thank you very much, Professor Lötvall, for your presentation. Apologies for rushing you a bit.
Sorry for, yeah.
No problem. Thank you very much for your time. And we can move on to our second speaker now, Dr. Dhanu Gupta, who, as I mentioned, is standing in for Dr. Samir El Andanusi. Dr. Gupta, the floor is yours.
Thanks, Charlotte, and thanks, Jan, for a great lecture. So basically what I'm going to talk about today is about how we are actually using EVs for various therapeutic purposes. And what we are mainly interested in engineering them with various biotherapeutics and delivering them to different tissues for therapeutic purposes. I mean, Jan gave a really nice introduction of exosomes, so I think that I can skip. And so I can just directly jump on to what we're actually doing with extracellular vesicles and how we're engineering them for biomedical applications. Before I start, so just conflict of interest here, I'm a shareholder and consultant for Evox Therapeutics, which develops engineered EVs for treatment of diseases. So if you look at the therapeutic EVs, you can broadly classify them into 4 different fields. So the first ones are the native EVs, which are where we actually use the inherent abilities of EVs for therapeutic abilities. Most of the work has been done with mesenchymal stem cell EVs. They have been shown to have therapeutic effect in a lot of autoimmune diseases, and have also been used in clinics and in treating a patient with GVHD. Then is the new class of EVs, which are hybrid EVs, where we actually take an extracellular vesicle, unengineered, and then combine them with another synthetic delivery vector. It could be a lipid nanoparticle, and fuse them together to make a hybrid EV out of it. And then the two different class of engineering EVs, which we are working on, is one is exogenous engineered EVs and another one is endogenous engineered EVs. I will go more deep into what is actually about engineered EVs is. So when we say about the endogenous engineering, what we are actually doing is that we are introducing a genetic construct into the producer cell. And what it does is that when you have, let's say, a protein of interest and we fuse that to an EV sorting domain, it directly gets shuttled into the EVs. And this is what we have been and others have been using for decorating surface of the EVs with targeting peptides or load biotherapeutic inside the EVs from varieties of different proteins. When it comes to exogenous loading, what you do is you purify the EVs from the cells, and then you load them with your biotherapeutics. So either it could be by a pure co-incubation or by a chemical treatment or by electroporation. You can load like from small molecule drugs to RNA species, from oligonucleotides to a big mRNA and plasmid DNA. People have loaded using electroporation. You can also load proteins on the surface of the EVs like that. So what we're going to talk about today is about the two different parts of engineered EVs we have developed until now. So the first relates to the studying the biodistribution of engineered EVs with a really high resolution. And then the second part relates to the surface display of biotherapeutics and how we are using EVs, exploiting EVs to display surface ligands on them and to decoy biologics in vivo. So if you look at the exosome workflow overview, you basically have the cells. It could be an engineered cell here, and then what we do in large scale production of we culture them large scale, then we harvest the media, and then we concentrate them because for biomedical application or in vivo experiments, you really need liters of media because you want to really dose high as possible. And then what you do is like you concentrate that media, so by using a tangential flow filtration, and then you can get a concentrated exosome solutions. This is not purified yet, but it has also like other proteins and also contains exosomes in high concentration. Then we move forward by purifying these exosomes. We basically use a size exclusion column to really get rid of most of the secreted proteins and really get pure EV population in our prep. And then we do more EV analytics. We basically run NTA to just check the size distribution of these exosomes if they're in their size range of 50 to 200 nanometers. And then we run more characterization using a bead-based system where we look at 36 different surface marker on the surface of exosomes. And you can also look at the soluble protein inside the EVs by looking at the Western blot and really looking at the contamination marker like galaxin or GM130, which are the markers for apoptotic bodies, just to make sure that you don't have apoptotic bodies in your preparation. And then you can also we also run electron microscopy to really see the morphology of these exosomes and the purity of them. So to start off with, what we initially did was to actually evaluate how we can best engineer the EVs. So for this, what we did was that we selected the top highly ranked protein in the HEC EV proteome, which we ran. And we identified 14 different candidates, and then we genetically fused them with a GFP molecule and overexpressed these genetic constructs into the producer cells, harvested the EVs, and looked at by different analytical tools how much loading we get and how much percentage of population, or like exosome population, we can actually label by using this genetic engineering strategy. And what we saw was that on the right hand side, you can see we ran basically GFP molecule per vesicle using fluorescent correlation spectroscopy. This was done by Nicole Meisner in Novartis before. And what we saw was that when we overexpress these constructs into the producer cells and harvest exosomes from them, tetraspanin mainly seems to enrich the most GFP into the EVs, where we could actually achieve up to 60%, 60 molecules of GFP per EV. And overall, if you look at the percentage of EVs which are labeled, you could see the tetraspanin, like especially CD81, CD9, and CD63 seems to work quite well for us. Then what we did was that to further add on to this, we used this tetraspanin CD63, which was highly ranked in our screen, and then we fused them with different luciferase enzymes. And the reason for fusing them with different luciferase enzyme was to really use engineered EVs for biodistribution tracking. Previously, we have utilized lipophilic dyes, and the problem with lipophilic dyes is that they label basically all the population of exosomes, and secondly, there's a high risk that you get membrane transfer of those dyes. So you're really not looking at the true biodistribution of the EVs. And also it's important, like in therapeutics, that you're really looking at the population you have engineered and how does that distribute body-wide. Because when you even do the surface engineering, you may get like 10% population engineered. And when you use a lipophilic, like a journal staining, you look at all the 100 exosomes rather than looking those specific 10 exosomes. So you may underestimate your targeting abilities or delivery abilities of the EVs. And what we did was that we screened 5 different luciferase enzymes, and what we identified, like NanoLook and ThermoLook being the most efficient in loading into the EVs. And NanoLook was so efficient that we could actually see that by a naked eye using like a handheld mobile camera just after adding the substrate. And we use these NanoLook EVs to really quantify, do a proper pharmacokinetics of the EVs in vivo. So what we did here is that we injected animals with NanoLook labeled EVs and looked at different time points. And to our surprise, what we saw was that the EVs were actually taken up within 5 minutes after being injected. So we saw the highest level of accumulation in different tissues at 5 minutes. And what we saw was that rapid degradation of the NanoLook or the EV cargo right after injection. So what we got to know after this experiments was that the half-life of EVs were basically in plasma was basically 2 minutes, and everything is just basically taken up within 5 minutes after being injected. And then we moved forward just because we wanted to know more what is the fate of the EVs since they're taken up so rapidly. So what we used was that we used a ThermoLook by which we can actually do non-invasive imaging of the animals. And what we did here was that we actually injected the EVs and visualized them right after injection, what actually happens to the EVs. And to our surprise, what we saw, right within 30 seconds, they started accumulating in lung, liver, and spleen. And if you can see, the trend was like that it started first in lungs, and then it moved more to a liver, and then the spleen started lighting up at earlier time points. So this clearly shows that, I mean, within like injecting the EVs, like right after injection, the EVs are taken up rapidly in vivo. And what we also saw is that there was no major differences in the biodistribution trend between 2 different cell types like MSC or HIC. So we wanted to understand like what is really like if there's any actually differences in the tissue itself. So we did a high resolution mapping of the cells which actually take up EVs in liver, spleen, and lungs. And here we, for example, we have taken 2 different exosome type, and what we saw was that 2 different exosomes differ in the target cell types in those specific organ. Even though, let's say, you get like 60% accumulation in the liver, but 2 different exosomes actually enrich in 2 different cell types within that specific organ. I mean, especially in the liver, the EXO1 primarily enriched in Kupffer cells, where EXO2 was enriched in hepatocytes. And also with the spleen, we saw that the profile of the cells being taking up these EVs were totally different within the spleen as well. So coming to the next part of the talk, where we have actually used EVs for more therapeutic purposes. So what we have done here is that we have engineered the surface of EVs with cytokine receptors, which cannot really signal. And by this, we can actually decoy those pro-inflammatory cytokines in circulations. And we started this project by basically started screening for different surface engineering strategies, which we can actually use to display these cytokine receptors on the surface of the EVs. So we generated like batteries of constructs for both TNF receptor and IL-6. So both TNF alpha and IL-6 are highly pro-inflammatory cytokines, and they're clinically targeted in various autoimmune diseases. So that's why we decided to go for these targets. And we generated batteries or constructs from having different EV sorting domain attached to them in the N-terminus, and we looked like what could be the best strategy to actually display these receptors on the surface of the EVs. So what we identified was the syntenin being the lead candidate in our screen. And what we also saw is that if we actually start introducing like oligomerization domain in our constructs, we start getting much better activity in our reporter assay. So if you look at the bottom left, what we can see is a TNF alpha assay. And when we add our engineered EVs, you could see that you can downregulate the TNF alpha induced NF kappa B activation in the reporter cells. And if we compare two different EV designs here, you can actually see that when you have the TNF receptor with the oligomerization domain and a syntenin, it works far much better than having just without an oligomerization domain and only syntenin. And we did similar thing even with the IL-6 receptor here, where we actually engineered them with having another oligomerization domain. Here was a dimerization domain because these receptor dimerize in their native conformation. And what we could see is that whenever we have the dimerization domain, these surpasses the normal like monomeric designs. And then we created like stable cells out of this because transient transfection can create problems over time. You can also have the carryover of the plasmid DNA, which can actually affect your results. So we decided to go forward with the stable cells. So here we generated two different stable cell lines. One was with HEC and one was with MSCs. And you can actually see that the activity was kind of similar with transient transfection, and the activity doesn't differ so much between the cell types as well. And then we actually started looking at targeting STAT3 inhibition in muscles because STAT3 inhibition, STAT3 is a key regulator for muscle differentiation. So its downregulation is important for muscle differentiation. And what we did here was that we took these engineered EVs and added on the muscle cells. And what we saw is that we could actually downregulate the STAT3 phosphorylation. And as you can see on your left-hand side, and if you see on the right-hand side, when we treat the muscle cells with these engineered EVs, you can actually see the differentiation pattern changes totally as compared to if you look at the wild-type EVs. So we took it further and we actually tested this in a Duchenne muscular dystrophy mice model. And what we saw was that we could actually downregulate the STAT3 inhibition in vivo by just treating the animals with these IL-6 decoy EVs. And we can actually downregulate in the muscles, which is a really big achievement. So then we wanted to also compare this strategy to what clinically is available. And what we did was that we compared these biologics to clinically approved biologics, and what we could see is that when we compare our TNF binding EVs with etanercept, which is a clinically approved soluble TNF receptor, we could actually see that these EVs were basically log better in IC50 values. And if we compare them in vivo in a sepsis mice model, what we could see is that engineered EVs with TNF binding ability surpassed the clinically approved drugs. And then we also tested these EVs for targeting multiple sclerosis, is another inflammation model where you get neuroinflammation. And we treated the animals, like we did multiple treatment of the animals with these IL-6 decoying EVs. And what we could see is that we could actually control the clinical score in these animals. Whereas, I mean, we also, apart from IL-6 and TNF alpha, we also engineered these EVs with IL-23 binding cytokine receptors. And what we could see is that even we could control of this IL-23 induced pro-inflammation, multiple sclerosis. And if you do a post-treatment, we could actually reverse the clinical score in those animals. And after that, we wanted to create more like multimeric decoys. So we were more interested in if we can actually have two receptors being displayed on the same EV surface. And so we co-engineered the cells with both of these receptors, and we checked if their activity gets affected by having both receptors simultaneously. And what we saw was that it was kind of similar to having single decoy over having both double decoys. And then here's just an example of this co-engineered EVs. And what we could see is that when you have the co-engineered EVs, the body weight of the animal goes much better. And if you look at on your left-hand side, this is a video where animals were treated with decoy EVs. So on the right-hand side, you have the animal which was treated with the decoy EVs. And you can see that the tailgate is kind of normal of that animal. And if you look on the left-hand side, this is an animal treated with control EVs, which has like this paralysis on below the abdomen. So the next part it's one of the other project we have started working on is on the antibody decoration of the EVs. So what we are doing here is that we are engineering the producer cells with tetraspanin, which have an FC binding, antibody binding domain on their surface. And when you get these EVs, these EVs can actually bind to the antibodies on their surface. And here's more like an analytical tool where we can actually show that that these EVs are actually indeed binding to the antibodies. So if you look on your top left graph, this is more like a run on a flow cytometer image stream, and where you can actually see when you have the engineered EVs with the FC binding moiety on them, they can actually bind to the APC positive antibody, whereas the control EVs do not have the ability to bind to these antibodies. And then we have also tried them to actually induce trigger uptake into in the cells. And when we add a target like a receptor antibody on the surface, and we could actually trigger the HER2 triggered uptake of the EVs into the tumor cells. And here's more example we have tried with different antibodies and different cells. So here's an example in a melanoma context where we actually target PD-L1 on the surface of these cells. We took these antibody binding EVs and labeled them with PD-L1 antibody, and we could actually trigger uptake in melanoma cells where the native exosomes without the antibody binding capabilities do not have any uptake in these cells. And then we did the same with again with the HER2 targeting as well. We could actually see that when you have the engineered EVs with the right antibody, you can actually trigger the uptake into those into those cells. Then we looked at the more in vivo, if we can actually enhance the uptake using with this. And what we saw was that when you have the PD-L1 antibody, you can actually target the EVs in vivo in tumors and in the lymph nodes. Whereas if you don't have the antibody, you do not get so much enrichment in those tumors and the lymph nodes. And here's another example where we have used transferrin, which is transferrin receptor, which is highly expressed on the surface of brain endothelium. And by this, you can actually target the EVs even to the brain. So that's pretty much. I would like to thank Samir and all the other team at KI and the team at Matthew Wood in the Matthew Woods lab, especially Mariana, for all the work and the help for these projects. Thanks.
Thank you very much, Dr. Gupta. So same goes, if you have any questions, please post them in the Q&A box. I saw that Professor Lötvall has answered Maria Enrique's answer. I'll give it a few minutes. If you also fancy asking your question live, you can raise your hand and I will allow you to talk. Okay, so we have a question here. Oh, it's Professor Lötvall. I'll give you the microphone.
Yeah, I can see the question. Yes.
I can unmute my microphone. Yeah. So
yeah.
I'm really curious about these decoy vesicles, and they're quite exciting, of course, because if they can function like neutralizing antibodies, that would be fascinating. Now, the big difference is that the neutralizing antibodies are probably circulating for a longer period of time, and vesicles are highly taken up by different cells and processed and so on, including the liver. And could you comment on how often you would have to give these to sepsis or whatever?
So I think it was a great question. So as I showed you with the when we saw with the NanoLook that the EVs have a really short half-life, and so I would assume that these decoy EVs are in circulation for probably like 2 to 3 minutes, or So majority of the EVs are cleared between 2 to 3 minutes. And regarding the dosing, I would say that you may need to dose multiple times, depends what context it is. For sepsis, I think since you get a storm, cytokine storm, I think one high dose could be useful. And the beauty with, I think, with EVs is that it's not shown by us, but at least by others, that when you have a more pro-inflammatory scenario, these EVs actually enrich in those target tissues. And that's why I think we believe that that's the reason that these EVs are more superior in activity as compared to the other biologics, which do not have the ability to home into a specific tissue.
My bias is that the liver is very active in sepsis as well, and maybe your vesicles are actually having activity in the Kupffer cells and macrophages or what have you in the liver itself. So is that possible, you think? Could the vesicles actually mediate biology even though they're taken up by a macrophage-like cell?
So, I mean, it's hard to say, like if they will mediate the biology or like if they can actually work when they're taken up.
Right.
It's hard. It's really hard to prove it, and it's hard to say that if it's possible to actually happen like that. So what I can, I mean, we also tried to see if the receptor shuttles back. I think we didn't see so much the receptor like being taken up and shuttling back on the cell surface, which could be one explanation why you could have more longer effect, because then you would have the cell expressing those receptors on the surface. But we didn't see that either. So it's hard to say. So my guess is primarily it's happening in the circulation or in the local tissue interstitial fluids.
Thanks.
Yes.
Then Benedetta has asked, what is the rationale for the increased effect of decoy receptor carrying EVs? Is it a matter of concentration of receptors carried by the EVs or acting at the same time?
So I think the rationale of increased effect, I think is more matter of concentration, like how many receptors you have carried by the EVs, I would say. And if I understood your question correctly, so I mean, we also want to lower down the dose of the engineered EVs because I think producing them is a tedious process. So we wanted to have more receptors, so we, as much as possible, so we can actually lower down the dosing and the production, like the load you have. If that was the question, if I answered right, I mean, Benedette, is that what you're looking for?
Yeah. Actually, my question was more related to the comparison of the decoy receptors on EVs versus those as soluble forms.
Okay.
Just to understand whether it was really something different, like aggregation of receptors on the cell surface, or simultaneous effect on the same cell, or I don't know.
So I think if you compare that to soluble receptors, so the reason they are far more active is that because, as I showed you earlier, like when we run fluorescent correlation spectroscopy, we can actually get 40 to 50 molecules of those GFP into that one same vesicle. And by this, I think when we engineer them, we get 50 different receptors and even 50 to 100 receptors on the EV surface. And that's why they're far more superior as compared to the single biologic, which has only one receptor.
Mm-hmm. Okay, thanks. Yeah. That's clear, thanks.
Okay, well, if that's it, if there aren't any other questions, I think it only remains that I thank both Professor Lötvall and Dr. Gupta for your time, your effort and your presentation. They were both very interesting, and as a reminder, these will be posted as soon as possible on the conference webpage. So for those of you who missed a bit or who want to go over it, you can go and watch it again. Thank you very much.
Thanks,
everyone. Take care and hope to see you for the next live sessions.
Thank you. Stay safe, everyone.
Yeah. Bye, guys.
Bye-bye.